Terrific. It's my true honor to be here, as I stated before. What I'd like to do today is talk about my pursuit of understanding eosinophilia and really focus in on the types of research that was advanced based on the wisdom and guidance that K. Frank gave to me. Frank was my PhD advisor, and he subsequently was my long-term mentor. And I'd say similar to what Josh mentioned, that our relationship did grow into a friendship. This picture shows me as a PhD student on the left. On the right, 30 years later. What is interesting is that Frank looks the same in both pictures, pretty much. In the photo on the right, I am Frank's age in the picture on the left. Now, if you are a keen observer, you will notice that I'm actually wearing the same tie in both pictures. Now, Frank didn't just talk the talk. Frank walked the walk. And what I mean by that is that he devoted his time and energy to truly meaningful and impactful science and providing sage advice. This is illustrated in this photo when he made the effort to visit Cincinnati Children's Hospital for several days to serve as our external scientific advisor. Another example of Frank's dedication and zest for life was when he accepted a speaking engagement in Israel at the age of nearly 90. He told me that he did this in large part to learn about Israel and to spend time with my family and I. One of Frank's pastimes I learned was estimating the time of the sunset. This photo captures the bet that we had on the Tel Aviv beach when he bet us at the time that the sun would set. As usual, Frank proved to be the winner. My research with KFA started with the fundamental observation shown here. This was that the peripheral blood eosinophil was in fact a long-lived cell. This was contrary to the conventional wisdom at the time, which had indicated that eosinophils died soon after entering an inflammatory site. KFA's response to this initial finding was shown here. Don't answer a question that I didn't ask, because this experiment was actually meant to be a negative control for another experiment that he asked about, which was related to basophils. Nevertheless, Frank let me pursue this in his lab and eventually proving the theory that provided the basis for a new paradigm that eosinophils were long-lived cells that appeared to be tissue-resident cells, not just peripheral blood cells. In the early findings of this set of experiments, we speculated that eosinophils were, that this system was relevant to tissue eosinophils, as these long-lived cultured eosinophils had acquired new phenotypic post-mitotic properties related to their hypodensity, enhanced leukotriene production, and cytotoxic activity. We soon went on to characterize the eosinophil poietins and proved that one of them, IL-5, was contributing to human disease. In the shown experiments on the left, we first demonstrated a dose-response increase in the eosinophil survival activity of serum of patients with hypereosinophilic syndrome. We then showed that an antibody against IL-5 blocked this activity. These findings were indeed driven by KFA's principle that if it doesn't dose response, it's not science.
If you're a business owner, then animated explainer videos are the best way to capture attention.
Based on these collective findings of my research and others, we proposed a model for the regulation of eosinophilia in this New England Journal article review. Of note, we emphasized the importance of tissue chemoattraction signals in addition to the role of IL-5 primarily in the bone marrow. Our research on this subject started off when we had cloned the eotaxin gene, initially in guinea pigs, based on an N-terminal sequence provided by Tim Williams' group. The figure shown here is an initial observation of the pattern of the eotaxin expression in the guinea pig based on northern blot analysis. The surprising point about this data was the interesting constitutive expression of eotaxin in the lung and intestine, the two tissues that harbored eosinophils under homeostatic conditions in guinea pigs. This finding provided the first molecular insight into the molecular regulation of tissue eosinophils. In order to prove this theory, we subsequently knocked out eotaxin gene in mice. The results demonstrated an essential homing signal for tissue eosinophils. As the primary reservoir of eosinophils, the intestinal tract was nearly devoid of eosinophils. We went on to quantitate the level of eosinophils in the graph on the right. And please note that we published this graph without statistical analysis, no p-values. And this was based on Frank's motto, which was that if you need statistics, it's not real science. We went on to further prove this theory by demonstrating that eotaxin indeed had a role in regulating allergen-induced eosinophilia in the lung. We first showed that administration of eotaxin, which is in the background, and these are animated slides, unfortunately, but the image in the back is basically sheets of eosinophils that we found after we instilled eotaxin into the mouse lung. And we noticed it's very rapid and eosinophil specific activity, which was enhanced by IL-5 overproduction. Later, we had identified eotaxin 2 in mice, and we generated mice that were deficient in both eotaxin 1 and 2. And we showed in this graph that there was a 98% reduction in allergen-induced eosinophilia. After reviewing this data, Frank indicated that I'm now convinced that you have uncovered the molecular identity of a long-pursued eosinophil chemotactic factor of anaphylaxis that Frank had been working on with his colleagues for a number of years prior to these findings. A major focus of my research has been to understand eosinophilic esophagitis, not only because of its strong unmet need, but also because the disease is characterized by profound tissue eosinophilia. Furthermore, and importantly, the disease provided me with an opportunity to study human tissue eosinophils derived from routine endoscopic biopsies. Basically, EOE provided a bonanza source of human tissue eosinophils in the context of allergic inflammation. We have taken multiple approaches to understand esophageal eosinophilia, including a focus on the molecular analysis of esophageal eosinophils in murine models of EOE. Two recent approaches include single-cell sequencing, shown above, and ATAC sequencing, shown below. The latter approach, the assay for transposase accessible chromatin, permits a first-pass understanding of chromatin structure and accessibility in eosinophils. In particular, bar-coded transposases are integrated into accessible chromatin, and next-generation sequences used to map the genome-wide epigenetic landscapes. This slide shows the data from single-cell RNA sequencing of the mouse esophagus following an EOE model. One can see that eosinophils are primarily composed of two populations based on this analysis, and these cells have been further characterized by the ATAC sequencing in the next slide. Panel A in this slide shows the complete separation of hematopoietic eosinophil, bone marrow derived, from tissue eosinophils based on their chromatin landscape. Panel B depicts this data in a heat diagram. These collective findings are quite striking because they show very strong separation between the eosinophil epigenetic landscape as a function of the eosinophil population. We use this information to uncover the potential functional role of eosinophils, as shown in the next slide.
Helen Hansel was able to win any competition she took part in. She was known as the contest queen.
Esophageal eosinophils are functionally enriched in pathways related to inflammation. which you can see in the circles, compared to the hematopoietic eosinophils. These eosinophils are very enriched in sensing response to external signals, TLR signaling, response to bacteria, response to LPS, for example, and apoptotic regulation consistent with the importance of survival in inflammatory sites. In contrast, bone marrow hematopoietic resident eosinophils are involved mainly in homeostatic metabolic processes. We use this epigenetic landscape information and RNA sequencing to map out the transcriptional factors involved in regulating tissue eosinophils, as shown here. In panel A, the predicted motifs are shown. Panel B highlights the enrichment of select transcription factors in esophageal eosinophils. Panel C shows the predicted interaction complex of the implicated transcription factors. And finally, in panel D, we show the ATAC peaks that share motifs for select transcription factors. What's shown particularly in these figures is that there is a remarkable conservation of an AP1 complex of transcription factors. Notably, these are uniquely different than the transcription factors that regulate eosinophil development. We next aim to prove that the role of tissue cues and select transcription factors in instructing the acquisition of tissue eosinophil phenotypes. In the upper panel, eosinophils were co-cultured with esophageal epithelial cells in the presence and absence of IL-13. This resulted in upregulation of relevant genes in eosinophils. Of course, these are just representative of many. This lower panel tests the role of specific transcription factors through collaborative studies with Annalise Pisalia and her mentor Kate Quinlan in Australia, where eosinophil cell line EO1 is knocked out for some of the transcription factors that are in the AP1 complex that we've identified. This includes JUN1 and ATF3, and the results of single knockout as well as double knockout are shown. And one can see that JUN has a generally positive effect, whereas AT3 is mainly a transcriptional repressor in eosinophils, and the knockout of both reveals cooperativity. So I'd like to conclude today in terms of the research that I presented with you with an overview of eosinophil development and tissue specialization. Our studies have identified IL-5 as a key developmental cytokine and eotaxin as a chemoattractant involved in eosinophil homing during homeostasis and inflammation. Furthermore, I have shared the concept that the tissue eosinophil is instructed by local environmental cues operating through distinct epigenetic and transcriptional programs, which are distinct from those involved in eosinophil development. I'd like to thank all the people that contributed to this work, which involves a group of diverse researchers coming together, including GI experts, research lab people, allergists, pathology experts, geneticists, and other investigators, as well as support groups, particularly the Curative Foundation, the SIGA group collectively, which is supported by the NIH, as well as the various NIH institutions and family foundations that have supported this work. I'd like to close by thanking K. Frank Austen for accidentally introducing me to the eosinophil, for his long-term mentorship, for teaching me about high-quality science, and for demonstrating, as John mentioned, the importance of family, for teaching me to live life to its fullest. And I will stop here, or I will break one of KFA's cardinal rules, which was, quote, to cut out the editorials and just give the data. Thank you very much for your attention today.